so we got passed through that, and today I'll be talking to you about some of our work in foundation models for science, mostly focusing on learning on molecular data. And I think, at the, we are on the second day of AMLD GenAI event, and to this audience, the word foundation model should not need any further definition. One thing I want to still highlight that while we all see that the foundation models, when you're talking about them, we mostly focus on transformer or similar architectures. Actually, they could be, you can think of a more of a generalist vision of foundation models, while we are still conforming to the notion of pre-training on a large amount of data sets of different modality and using some form of self-supervised training, they need not be transformer. And actually, we have found that even before the transformer emerged as a main frame architecture, models that were predate to transformer like LSTM and all, they actually benefit equally from training on large volume of unlabeled data through self-supervised or some form of unsupervised training. But at the end, you get this generalizable and adaptable data representation, which then can be adapted, fine-tuned, transferred to different downstream tasks. And we already have seen its benefit on different tasks associated with language, on medical imaging, what else? I think robotics, and quite a few. And at IBM Research, we are tapping on many of these applications of foundation models. So we are building new theories, algorithms, and methods for training, learning these models. And as you see that we, while we, of course, all of us were amazed to see what was happening with language, but soon we see that, the same notion of learning a large model on massive volume of data is not necessarily limited to language. We are at IBM, we are looking into sensor data, speech, AI for code is a big effort. I urge you to look into there if you are interested, as well as digital interactions and chemistry and biology and scientific domains and as such. So why we think that, that why this same notion of learning is now working across different domain, different scientific tasks and beyond. Because if we were looking into, again, predate this term foundation model was coined, we would see that, vision and language and speech, these were kind of, segregated community, and they had their own favorite architecture. And what we see with this, storm of foundation model, that all of these communities are now emerging together and sharing more than ever before, that the innovations that are working in for one task, one modality, one particular domain can be utilized in other. So one basic thing that is happening in all of these different domains is that this notion of foundation model, transformer-based training, is actually identifying the structured learning that is underlying in each of these domains. So if we are like what I'm showing you here in this chart, that in language, you can see this hierarchy that is building up the way where we are seeing some linguistic phenomena happening here. Not cognition, though, I think we are yet to get there. But we could see that there is some structure, syntax, semantics that are all emerging and giving access to this linguistic behavior, which is modeled by large transformer like models. The same thing can happen when we are talking about learning on chemical data or biological sequence data, where atoms are constituting the molecules giving rise to certain properties, and then we see phenomena emerging that are like governing chemistry and biology and more. Actually, the same parallel can be drawn to finance data, and this is not just a picture for the sake of picture. Indeed, we have trained similar to GPT for financial data and BERT for financial data, with the goal of user behavior prediction and with the goal of user behavior data generation. So today I'll be, yeah. So what I wanted to again stress that in many of these cases, though we are building on the notion of learning from language, but actually the data could be multimodality. And again, molecular data is one wonderful example of that, where you can get the data in the form of time series, knowledge graph, language, image, 3D geometry, and so on. And then our goal here is to train a foundation AI model for some application. either it broad application, either could be for biologics, for chemical, for finance, or for climate, while we are trying to build this model that would be then adapted to define downstream tasks where the data is often small compared to the pre-training phase. And then we'll be subjecting the same model and adapting it to define downstream tasks, be it design, optimization, causal discovery, and so on. And my group at IBM works on not necessarily on a particular domain, but where we work is this workflow and tools, where our goal is to develop new pre-training workflow, new architecture and algorithm for control generation, for architectures for geometric and causal neural nets, and so on. And we work with the hardware, the middleware team, as well as the experts in the domain to deploy such models for some real use. Now, if we are now focusing on the scientific discovery and workflow, particularly the scientific discovery and workflow that require learning from the molecular data, I think many of those design workflows or the discovery workflows can be mapped in a paradigm where we are talking about design of candidates, and then we want to gather some evaluation, multi-tier evaluation for those proposed candidates, learn some feedback on those proposed candidates, and then adapt to the model or to those proposed feedback in an iterative manner. What is interesting here that no one in, if you talk with someone in chemistry or biology lab, no one wants to take a look at any generation, any design that's coming out of a generative AI workflow or a generative AI model. The first thing they would ask that, that what are some of the criteria that you have used, and if they are meeting those attributes that are the minimum requirements for synthesizing and testing such a proposed design at lab. So what is very important here is this notion of controlled design, controlled discovery, or this guided generation. And you can frame it in many ways. You can, algorithmically speaking, you can come up with new controlled sampling algorithm, constraint optimization, or you can do active sampling. You can, of course, generate a lot of new designs and do some forward scoring on screening, and also do cross-modality learning. So we have been working on many of these aspects over the year, and in the remainder of my talk, I hope to give you a glimpse of some of those models, training paradigms, and also show you some real proof that if they work or not and where they fail. Before I get in there, we of course work, though we are part of industry research lab, we work with the community, and we want to open source as much as we could. So one of our recent development that we call mole formats, which is a foundation model for chemicals, we open source that through an UI in this website, moleformat.res.ibm.com. What is lying behind mole formats UI is the largest model that we have trained so far. I'll talk about that in a bit. And we have 4 different operations available on this model. One is screening, one is search and browse, generation, and model explanation. Screening, as it says, that we are providing some predictive model on top of the pre-trained model, like toxicity and so on. Search and browse is more like Google search, but here we are using the mole format encodings to come up with the nearest molecule from the database. Generation is showing some generation proposed design, and model explanation is giving access to the model attention. So now, like an expert can look into that attention and see that is the model just predicting for the sake of prediction, or the model is learning something explainable, something meaningful, which is coming across from the model attention. And anyone can access this UI. This is being hosted using cloud, and we just need a valid email to get you on and get you to try it. So what is mole formats? Mole formats is the foundation model, as we define it, is the foundation model of small molecules. The small molecules here are coming from Babchem and zinc data. When we trained the model, this was published in Nature Machine Intelligence last year. I think to date, it's still close to the model that has been trained on largest amount of chemical data. The model size is about 50 million or so. And what we in terms of training data size, it is trained on 1 billion chemical smile string. Though we are from IBM Research, and you might think that we have a lot access to a huge number of GPUs. Actually, when we are training the model And given our focus on natural language, we didn't have had access to a lot of GPUs back then. And as someone says that, sometimes constrained resources are actually the mother of innovation. So to that extent, we came up with adaptive bucketing and some other parallel training techniques to make the model be trained on 16,300 GPUs. And it took us only, I think, 5 or so days to do 4 epochs on this 1 billion chemical strings data set. Now, as a result, what we get is this Molfarmer Excel model, which then we tested on many different benchmarks by fine-tuning the model on those different downstream tasks. And this task could be like some predicting some quantum mechanical property, like going back to the benchmark like UM9, and all the way up to looking into like blood-brain barrier permeability prediction, toxicity prediction, and so on. What was the interesting fact that, to similar to what we have been seeing like GPT or BART, like in natural language, Molfarmer indeed can perform very well across all of these different tasks, going all the way from quantum mechanical to physical to physiological. And to date, we have tested it on more than 70 plus downstream tasks, and the number of tasks are increasing as we speak. And some of the interesting fact that we observe here that I have to plug in my, my laptop. So. Okay, I believe we are safe. Okay, and when we compare the model, this model trained on chemical smile string, we see that the model is actually performing sometimes even better compared to models that are trained on molecular graphs for specific tasks, even for 3D molecular coordinates. So this was to some extent surprising because particularly for quantum mechanical tasks, it is believed that the structure of the molecule is very important. So you need to learn from that kind of highly specific structural information. And what we see that when the model is now being trained at scale in terms of model parameters as well as the data size, indeed the model can learn more when it is only being trained on the chemical smiles data. And here we are showing, some these tables show that how the model is doing on some regression task, and a number of classification tasks. And these baselines are some of them are supervised, and some of them are pre-trained. And you could see that the Molfarmer Excel, the largest model, actually performs consistently across all of these tasks. Now, even more interesting, so back then last year or the year before, then we were just getting to learn that these models, when you train them at scale in terms of data and parameters, the model learns something more than what it was intended to train on. And we just wanted to see if something similar happens for Molfarmer kind of models, because now the model is only being trained on chemical smiles data, which is just a linear notation of how a molecular structure should look like. So there is no three-dimensional information, and there is also no connectivity information in there. What we see that when we are looking into the attention map of Molfarmer trained on certain molecules, we do see that actually those attentions are correlating with the spatial distances between the atoms in the 3D space. So this was something surprising to some extent, because again, the model didn't have access to any spatial information when it was being trained. And we do see that this phenomena only emerges when the model becomes bigger and when the data size is hitting this billion plus scale. If we start scaling down, this effect goes away. We also saw that the encoding space or the embedding space of Molfarmer actually has some structure, and this structure is indicative of some preliminary labels or preliminary data annotations such as molecular test. What you are seeing here is the mapping of the Molfarmer encoding of different molecules that are annotated with different tastes like bitter, sweet, and sour. The interesting fact here is that during the model training or fine-tuning, the model never had access to such data. But when we are mapping this label information post hoc onto the encoding of the molecules in mole former space, you could see that now like the molecules are being separated that are bitter from the sweet ones. So again, the fact here is that now we do see that what we were seeing in language and other modalities, that when these models are being trained at scale in terms of model complexity and data scaling, we do see this kind of phenomena happening, that new structure is emerging, new information learning is happening. And to be honest, we know that this is an effect of scaling to some extent. Not everything can be emerged as a result of scaling, but definitely, some of the patterns that our model is learning, which is kind of hidden in the input data, is now being more and more apparent when we are looking into the model space. And as a natural transition, we of course, the mole former Excel is an encoding model which you can use for different predictive tasks or for molecular search. But for generation, now we have been working on mole former GPT. This is something that we are working on right now. It is again built on the mole former Excel as the encoder architecture. Now we are extending that to the decoder architecture. In terms of data, in terms of the model architecture, it remains the same. One fact I forgot to tell, that we in mole former Excel, we use linear attention to get rid of the quadratic complexity that comes with the standard attention mechanism, as well as we use something called rotary embedding, which is to encode or to enforce learning of the relative position information of the atomic tokens within a molecule. Again, we leverage such tricks in training of mole former GPT. And as a result, what we see as the first test, that this MFGPT model actually can generate valid, unique, and novel molecules, which are like the very first test of any molecular generative model. And what we are right now seeing, that what are the effect of prompting and so on, and can it also take like a fragment as a prompt and can generate a complete molecule that does have the fragment and is novel and so. So that is something that we are working right now. And so like I mentioned already, that any generative model is of no interest when we take the generation to a chemist or to a biologist. What we need to do is here control generation. And this is something we have been working even before we started looking into transformer, and for a few years right now. So once you have learned the latent space of molecules by using some model of mole former, right? So from there, we need to look into how we can guide the generation of the molecules such that it meets certain criteria like low toxicity, high activity, low cost of synthesis, and so on. And we have, over the years, we have now defined and implemented a number of such algorithms for control sampling and optimization. I'd skip going to the detail, going into the details. But what I want to show you that, of course, if you can look up the publication if you are interested, you can frame the question in terms of sampling, in terms of optimization. But I really wanted to get you into that, does this really work? Like, comparing your models across benchmarks are nice, but did any of your mole former model has been able to discover anything that didn't exist previously? And we have seen some success while we have done experimentation with the mole former proposed designs. Again, I must say that given where we are and given how the community works, definitely we didn't have a huge amount of resource to do such validation at scale. But to date, we have done validation at two different kind of use cases. One is for antimicrobial design. The reason we did it was an AI for social good project for us. And the goal there was that, can we accelerate the discovery timeline of new antimicrobials, given that, new antimicrobial discovery has been really, really a tough problem, and that is very much needed to tackle the antimicrobial resistance worldwide. And with this work, we show that when we do control sampling from generative model, and here what you see is the two new antimicrobial that we tested in the lab. So basically 20 new designs were sent to the lab, a lab in Singapore. 5 of them came out to show antimicrobial activity to at least to some strains. And then 2 of them, the one that are the highlights here, they did show broad spectrum activity across a number of strains, even the one that are multi-drug resistant. And we also found that those 2 actually showed in vitro low toxicity up to mouse models, which was really, really cool. And our experimental collaborator was super happy of the outcome. More so, one aspect we saw from learning from this large amount of data, that the molecules that were the best from this batch size of 20 did show properties such as low induced resistance and high potency against resistant bacteria. This was not actually included in our control sampling objective. Our control sampling objective was only considering novelty, high activity to different bacterial strains, and to the low toxicity. So this was like, again, some outcome that were not considered in the objective function. And we, being no domain expert, we were happy to see, but we had to ask our collaborator that why this was happening. And her answer was that likely the induced novelty that was considered in this kind of control sampling was giving rise to this very intended, very desired property of low induced resistance and high potency against resistant bacteria. This is something that, if we were to do like, some trial and error approach or some template-based design, the novelty is a fact that is often gets ignored or undervalued. And here, when we are doing this data-driven generation, we do see that the novelty can be something that we can be enforced, and definitely you can go as high as novelty without losing the other, functional attributes like potency and toxicity. And so at the end, the summary is that this sort of generative AI models for scientific discovery allows us to look into some part of this big landscape that has been previously unexplored, which we can use to our benefit and against the fight of this drug-resistant bacteria. Another validation that we did, and this was during COVID time, and the work was only recently published because the submission and publication timeline, there is a big lag in between. But anyway, in this case also the model that we trained, the generative model, didn't have any information about SARS-CoV-2. So it was trained on all information available that happened before SARS-CoV-2 emerged in the world. So what it was trained on, this model, trained on protein sequence relation, molecular chemical SMILES data, as well as the protein molecule, small molecule binding information. So this was all that this model was trained on, but no mention of SARS-CoV-2, any data with SARS-CoV-2. So now once the model was trained, during generation, you could prompt it with any protein sequence information. So what we did then, we prompted it with SARS-CoV-2, 2 different SARS-CoV-2 target information. One was the main protease and one was the receptor binding domain of the spike protein. What we found that, and then of course we ranked the generations and so on, and we sent them again to lab for experimental validation. And this work was done in collaboration with Oxford University, Dave Stewart's lab and Diamond lab in UK. We synthesized only 8 molecules this time, 4 for main protease and 4 for spike protein. Not because we only wanted to synthesize less, but this work was happening when the world was going through the pandemic, and then we're in Ukraine, where our chemical manufacturing company's headquarters is, so we were limited in terms of resource. But anyway, the fact here is that, out of these 8 molecules that were designed by this generative foundation model technique and tested, synthesized and tested in lab, 4 of them showed target-specific inhibition. More interestingly, 1 of them showed, broad spectrum potency. The model has only seen the original Wuhan sequence during the prompting during inference, but the molecule showed broad spectrum inhibition across up to Omicron. So all what I'm saying that, when the models are now being trained that are by learning this relationship that are embedded in this large scale data, the outcome of that is more general than before. And we do see, as a result that, this sort of broad spectrum activity is coming along. And also in this case, we found that a new binding mechanism. Because we gave the protein sequence as a prompt, we found that once the molecule was synthesized and tested, it's not binding to the regular interface of ACE2 and spike protein, but it's binding to a very new pocket of spike, which is more conserved than ACE2 and spike. So our collaborator Dave Stewart is very excited that if this is real, then we would be likely, coming up with new molecules that would be working against defined mutation, because it's targeting a new pocket which is much more conserved than the antibody binding pocket of spike. So, okay, so how many minutes I have? Okay, good. Okay, so far I think I have shown you that a foundation model for molecules, they work for prediction and for generation tasks. But the question now is that for every new task or for every new molecular domain, do I need to train a model from scratch? That means I have to have access to GPUs, I have to know how to build a model. Is that true? So here we are exploring a new paradigm which we call model reprogramming. And this might sound like black magic, but this is really looking into the new frontier of foundation model research. What we do here is that we take a model trained on natural language, and if you recall, at the very beginning of my talk, I said that all of these models are learning to learn some structure embedded in data. So the point here is that if they are all trained with the same purpose in mind, or somehow they are learning the same objective that are embedded in the data, can we repurpose, let's say, a model trained on English to protein sequences or to small molecules or something else? That is exactly what we do here. The goal here is to map the protein language to English by learning a dictionary mapping. And by label mapping, we are updating this dictionary mapping. So at the end, the protein language and the English language, and they are aligned. And all of this is done through not accessing the gradient of the model. So we are just looking into what is the loss, the cross entropy loss in terms of label matching, and then updating the embedding of the protein sequence, but not touching the English BERT model. And first we, of course, check that if this is working on different predictive tasks, and what we see that indeed it is working on different predictive tasks. But the advantage here is that the parameter efficiency, so as you can imagine, because we are not updating the main model, we are just updating some layers before and after, we are only learning a few parameters. So in terms of training cost is much smaller, the data need is also much smaller, and the performance wise in low data scenario, we do see that the efficiency of such model compared to supervised or pre-trained models are much, much better. And here we show the application in terms of different function prediction and structure prediction tasks of proteins. And we have also extended this paradigm to design problem, like you can think of designing new antibody by having an English BERT in the middle, and then learning some function before and after, so that you can now map the protein sequences that to something like English-like, and vice versa during the output. And we do see that such reprogrammed model indeed shows much better structural consistency when we are comparing with different baselines, but also giving rise to sequences generated that are much more diverse than before. And with that, I would come to the final slide, the slide before I list all of my collaborators. So I think we are seeing that large pre-trained models are emerging as a promising technology, not just in language, not just in problem related to vision, but also in scientific learning and discovery workflows. As a result, I think this is something really important, that we need very standardized frameworks for scaling and governing the uses of this model. Of course, this model should be used by the broader community, and there shouldn't be one or two players who are dominating the uses of them. But on the other hand, given that they have this power, and we do see the power across different discovery, different design tasks, I think it's really important to monitor how they are being used and have a repository where every instance of such use should be reported. Finally, the models here, though I focus on learning from data alone, we have found that integration with domain knowledge and physics at every stage of learning is important. And it is important to go beyond the current benchmarks and metrics. You have heard that the importance of evaluation of LLM. The same actually is important here even more, because even ourselves, we have seen that how the labels are being defined, and if they are not consistent across different databases, that changes the entire model performance. So with that, I would just end by saying that we need to go beyond what currently exists in terms of benchmark datasets and metrics, and work with the community so that these discovery instances I have seen could be reproduced again, or may not be, but we know where they fail. And with that, I would like to conclude by thanking the collaborators that I have been interacting, working together over the years at IBM and outside of IBM. I have been very fortunate to work with each one of them, and without them, this work would not have been possible. And thank you for your attention.
Thanks a lot for this exciting talk, and for all the different excellent work. We'll start with question. Who has questions for Payel?
Hi, thanks a lot for the talk. Amazing work. I'm really curious about the last application where you reprogram language models. Like, were you also surprised that this even works? And since it works, how do you interpret this? Like, is there some kind of common logic between language and...
Yes, yeah. So we were not surprised that it worked. We were surprised by its performance when we compared with the baseline, because our expectation was that it will be comparable. But I think it took even us some time to realize that why it is giving this ability to design diverse sequences, right? So that was something that we didn't expect to begin with, to be honest. And I think the logic behind there is that the English BERT, the original BERT that was trained on much bigger data, the model is big. Of course, if you retrain all of the parameters, then the data set that we are looking into working on in this new domain is not enough, right? So by learning this, embedding the parameters for this embedding layer before and after, we have now advantage to only update a few parameters without touching the big model, but the big model is still there, right? So that nonlinearity that is modeled by this big model is giving us this access to many diverse sequences. That's how we explain it. And we actually have, we don't have a proof per se, but we have empirical experiment that if you lower the model size of the fixed English BERT model, actually this effect goes away.
Yeah, I just want to follow up on that point. There's actually some work, it's pretty old now, but where they had taken pre-trained models, but they pre-trained them on music or gene sequences, and then they went to these downstream tests. Yeah. So this is like the reverse of that, right?
This is reverse of that, yeah. And actually, that's a very good point. I think it's learning on music, something like that, right? And it showed that you can get a model that is trained on just some nested grammar of completely bogus word. You just have to, enforce some structure. And once the model is trained on this synthetic data with some structure, the model can do really good on some language task and so on. So meaning that, if you are here, we are only focusing on learning to leverage the structure and knowledge built in that pre-trained model and transfer that knowledge to other domain.
More questions? This Anu has another question.
Okay, this is maybe a kind of a technical question, but you mentioned that you saw good performance on QM9 and QM8, and these are like tasks which you would think geometric measurement would work. I mean, the results you showed are pretty weak baselines. This is from MoleculeNet, and there's been a lot of obviously better work from there. Do you still think that holds, or is this a fluke from the weak baseline?
No. So what I showed you here is some baseline, but one of the reviewer, I hope it's not you, he asked us to go back and compare the Molformer with SNET, DimeNet, Geom, GCL, and so on. And we did exactly that. And his bet was that it will be at least 1,000 times worse. And We expected, that it's not going to be 1,000 times worse, but definitely worse. And we did find actually it is indeed worse on some of the QM9 targets like U0 and so on. But what we found, it was only 8 to 10 times worse. So meaning that you can push the model just by learning from chemical smiles to that extent without having access to any 3D information.
Okay, yeah, that's interesting. It's like a lot of on the QM9 task, a lot of the errors coming from one of the
those are the targets that we picked, the hard ones, not the easy ones, right? And that's where we found that actually the model was indeed behind, but not by a factor of 1,000, 8 to 10 at most, right? And those models, then we also benchmark them on the physical property regression benchmarks. The DimeNet, they actually do very bad on those. So because I think, the point is that those models were supervised on QM9 alone, right? And they were fine-tuned so much on QM9, they are now losing this general purpose. Whereas Mulformer is kind of consistent general purpose, and it may not be the model trained on quantum interactions per se, right? But it is showing the potential to get to there.
Sorry, when you say the physical tasks, you mean like solubility or something?
Like lipophilicity, those, yeah. And those are all in the supplementary of the paper.